Hello, everybody. Welcome to today's joint webinar series between GlycoNet and the ACS Carbohydrate Division. So it's my pleasure today to introduce our first speaker, Professor Al Boraston. Al and I have known each other for a long time, and we share a similar scientific background in that we both attended the University of British Columbia in Vancouver. Dr. Boraston got his BSc and PhD at the University of British Columbia. He did his PhD in what was then known as the Cellulase Lab with Professors Doug Kilburn and Tony Warren. And then he did a postdoc with Professor Gideon Davies at York University in the UK, where he learned structural biology. And he came back to Canada and established a lab at the University of Victoria, where he's been ever since. And during the time that Professor Boraston has been at the University of Victoria, he's been an EWR Stacy Fellow, and he held a Tier 2 Canada Research Chair for a decade. Dr. Boraston's research is in structural glycobiology, and in particular, he's very interested in carbohydrate interactions between enzymes and their substrates, and he's really been looking into molecular pathogenesis in a variety of systems. And he's going to tell us today about O-glycopeptidases, a project that we are working on together. So I'm very interested to hear what he's got to say for the project today. So without further ado, Al, the floor is yours.
Thank you very much for the introduction, Warren, and thank you for the invitation to speak today, and good morning, everyone. So I chose to talk about an area of research that we have been tackling on and off for about a decade. And the reason it's taken so long to do it is it's actually a really complex problem, and that's these O-glycopeptidases. And so really the highlight today will be about their complex recognition determinants. So I know it's a little bit strange, and some of you will actually have seen portions of this talk before, but I've never done it all together and never incorporated new data. But to really, get the feel for it, I need to start a glycobiology talk with a weird beginning on peptidases. Just to remind everybody that peptidases are enzymes that hydrolyze the peptide backbone of proteins, and this reaction is essential to all living organisms. Now, those of you who are familiar with carbohydrate active enzymes will be aware that they're very nicely classified into several different clans and families. And there is a parallel classification for peptidase families, which can be found on the MIROPS website. And so I'm just going to bring up my laser pointer here. And so the MIROPS database classifies peptidases in several different ways, and what's going to come up in this talk today is my preferred classification, that is the amino acid sequence-based families. So the thing about the substrates of peptidases, of course, is that they're frequently post-translationally modified, and this comes in all different kinds of flavors, of course. The one that we're most interested in is glycosylation, and the types of post-translational modifications in terms of glycosylation are varied, but we're going to focus on O-glycosylation today, and specifically the kind where we have a GalNAc residue that's alpha linked to a serine or threonine. And so this creates the core for what can be a fairly highly modified set of O-glycan structures. And this O-glycosylation is abundant in particularly mammalian organisms. So where these two topics meet is this concept of glycoproteases, and these have actually been known for quite a while. And the classic example is this O-glycopeptide endopeptidase, which is actually a bit of an enigma. It originates from Pastorella hemolytica, and it's been used as a tool for decades now to degrade sialylated O-glycoproteins. And it was originally classified into this Metallopeptidase family, M22. It's now a defunct family because this appears to be the only member of that amino acid-based family that had this particular activity. So it's a bit of a strange bird. Of course, there's some other examples, and I'm highlighting them here, that have cropped up over the years. And what's shared by all of these so-called glycoproteases is that they degrade highly glycosylated proteins. And so mucin would be the model example, where you have a protein with particular domains that are highly O-glycosylated, making these hairy chunks of protein. Now, in terms of these particular enzymes, the question that hadn't been addressed is what the molecular basis for glycoprotein selectivity was. Is it just that these enzymes tolerate the glycosylation, or does it really need to be there? Does it really want these? And this is how we kind of got into this area. But of course, when we're talking about the degradation of glycoproteins, there's a biological importance to it too. And I'm just going to highlight that here, focusing on the gastrointestinal tract, specifically the large intestine. So this is a highly colonized environment, lots of bacteria and other microbes in the lumen. And they're separated from the epithelial cell layer by a mucus layer, which has two pieces to it. The first is an outer mucus layer, which is loose and it's colonized by bacteria. And then there's an inner mucus layer, which is more dense and largely aseptic. The mucus layer is primarily composed of mucins, so these highly glycosylated proteins, and they can be membrane bound or they can be these gel forming soluble mucins. Now, the thing about this ecosystem is it's in a nice balance. So in this microbiota, we have a variety of different microbes, which in this particular example are just divided between fiber degrading, mucus degrading, and pathogens. So the fiber degrading ones are the guys that are good for us, and when we eat fiber, they proliferate and their population is maintained. And then we have these mucin degraders, which will actually degrade this mucus layer. And then we have the opportunistic pathogens. And under normal circumstances, this isn't a nice happy balance where this mucus layer is constantly being regenerated. However, when the diet changes and it gets out of balance, the mucus degraders can proliferate, thus resulting in erosion of the mucosal layer and eliminating its barrier function. And so this makes this environment now susceptible to the infection by the opportunistic microbes. And so there's this really interesting balance, which is based on this mucus barrier, which is largely mucins, of course. And so in terms of the glycoprotein destruction, we can consider that in the perspective of healthy degradation of these glycoproteins in the mucus barrier, or unhealthy. But we can also use our imaginations and expand this concept of glycoprotein destruction to other areas of the body. And so one example that immediately comes to mind is many of the molecules of the immune system have mucin-like glycosylation, and it can be destroyed by invading pathogens. And so this raises the question of, do glycoproteases contribute to these key aspects of host microbe interactions? And this is a question that's always in the back of our minds. And there's lots of data now that is being produced that actually supports this particular question. I'm not going to talk about any of that today, though. So we backed into this area sort of by accident, and the reason for this is one of our pet microbes in my lab is Clostridium perfringens, which is a normal inhabitant of the human gastrointestinal tract. It's just not a very abundant one, and it's an opportunistic pathogen that when things get out of whack, it can actually cause problems. And so we were studying this group of enzymes which all appear to target the complex glycans that you would find on, say, mucins in a gastrointestinal tract. Now, when we were surveying these, we were also looking for other potential enzymes with similar architectures. And what popped up are these two genes, which encode these proteins I've shown schematically here, which share some architectural similarities that you can see there, but don't have anything that's predicted as a carbohydrate degrading module in there. What they had are these things that look more like proteases. And kind of a long preliminary story short, what we actually found over a few years is that these enzymes actually have mucin degrading activity. And so around the time that we had a pretty good grasp on what those particular enzymes did, a group in the UK published a paper on BT4244 from Bacteroides theta iota omicron, which degrades mucin. And the real crux of this paper was defining this really large family of related proteins. So it's PFAM 13402, or the M60-like family. I think it's been renamed as Peptidase M60 now. But suffice it to say, there's a lot of entries, and our proteins fell into this family. Now, what this family shares is the majority of them are from host-adapted microbes. Most are multimodular proteins, that is, they have a catalytic domain as well as some other stuff on them. And there's several different Murops families that are incorporated into this superfamily. That same year, another paper was published on IMPA from Pseudomonas aeruginosa that seems to target these O-glycosylated surface receptors. So we've been working on this, and we decided as a set of models that we would expand to BT4244 and IMPA to get a better grasp on how these things, which appear to have glycoprotease activity, actually work on their substrates. And so these are schematics of the three proteins, and what you'll see is that they're all quite large. The top two, BT4244 and ZmpB, are multimodular. IMPA, there's no definable modules, but what they all share is this M60-like domain with the gluzinsin motif. And in here is the catalytic glutamic acid. So we simplified our problem by stripping it down to the catalytic domain to study that. IMPA, we couldn't strip down. We had to produce as the whole thing. But suffice to say that they all shared the expected glycoprotease activity that had been previously reported. And again, making a longer story short, much of the data pointed to a requirement for O-glycosylation. And so we then hypothesized that O-glycosylation is not just tolerated, but it's actually a key substrate recognition determinant. And we decided to approach this hypothesis by engaging the glycobiologists at NRC and then Warren's lab as well when he was at Ryerson. And our approach was to generate really defined glycopeptides, where we'd have the naked glycopeptide, and then we'd have these different common O-glycosylated structures on there. And we assayed the activity of our three enzymes by thin layer chromatography and mass spectrometry. And what you can see here is that we got activity for all of them on at least one glycopeptide, but not on the naked peptide. So this is strong support for a real requirement for the O-glycosylation. We subsequently revised this. We did find that BT4244 was active on these linear structures, but remained inactive on a branch structure there. Now, what the mass spec had to offer, and in addition to confirming this, but all three of these enzymes really specifically cleaved the peptide on the peptide bond immediately N-terminal to the site of O-glycosylation, all three of them. And so this created this really interesting picture of this enzyme that was set of enzymes that were really specific for O-glycosylated motifs. And so being interested in the molecular basis of how these things work, we drilled a little deeper by structural biology. I mean, we're a structural biology lab, and so this is, you know, the direction that we tend to go. And so these are the apostructures of these enzymes, and there's not a whole lot informative here other than to say that they all have this zinc metallopeptidase catalytic motif, as well as this thermolysin-like core where all the action's happening. So this was conserved amongst the three different enzymes. You can see that IMPA looks a little bit different, but of course it's a larger protein. The apostructures, of course, aren't really informative. And this experiment here was the one that really launched this project. And it was one of the ones that a postdoc does, and you're kind of wondering, is this really going to work? Because it was definitely a lob deep into the end zone. And so this was Liz that did this. And what she did was she had a drop of ZmpB with the crystal growing in it, and she literally added bovine submaxillary mucin powder to it. So basically turned her nice crystal drop into a snotty mess. And reasoning that, well, there's going to be some active protein in solution and it's going to degrade it, and hopefully the enzyme will pull out the piece of bovine submaxillary mucin that it wants. And lo and behold, much to my surprise, this actually worked, and it worked really well. And what we were able to clearly model into the electron density was this glycopeptide here, where it had been cleaved immediately N-terminal to this threonine residue. And so when we look at the interaction with the peptide here, this is entirely consistent with what you'd expect for a zinc metallopeptidase in terms of where the water is, the catalytic glutamic acid, and the zinc binding site, etc. That, of course, wasn't really what we were interested in. We were interested in the flip side of this, because this is the threonine where the glycan is attached. And so if we rotate this around, what you see is this glycan here and all the interactions with the active site. In particular, this central GalNAc, the core GalNAc, is making a set of interactions with residues in the, not directly in the catalytic site, but in regions adjacent to it. When we look at the surface of this, you can see that the glycan is snugly tucked in there with the sialic acid packed up against this phenylalanine 727. And so this is what really launched the project. And we continued this on by solving the structures of BT4244 and IMPA with glycosylated amino acids. Now that we had a better grip on this, we could simplify our problem a little bit and not have to make lobs into the end zone. And to try and summarize what we're seeing here is a set of conserved interactions with the central GalNAc or the core GalNAc. There, BT4244, where this O6 is, this area is closed off. It wasn't closed off in ZmpB. This is where the sialic acid would be. And this explained why this enzyme was only active on the linear glycans and not the branched one. IMPA, however, we knew from microarray studies that it interacted with these branched ligands and was also active on the alpha-2,6 sialylated versions of these. So there's this additional potential set of interactions out here with W685 that we weren't seeing in the crystal structures that we had. But altogether, we can interpret this in terms of what was known about protease active sites or peptidase active sites and how we have to rethink this with these particular set of enzymes. And so this is the really elegant model for a typical peptidase, where a peptidase active site has these subsites, which are termed the S sites, and they're labeled 321. We have the central here, and then 1', 2', 3'. They accommodate P residues in the substrate, and the point of cleavage is between P1 and P1', between the S1 and the S1' subsite. So a really nice, simple, and elegant way of visualizing these active sites. This, of course, didn't apply to our enzymes. We were looking at something that was a little messier, something like this, where we had specific recognition of the glycans. Our P1' subsite would always be a threonine or a serine, and the point of cleavage would be N-terminal to this, kind of adapting this to what we were looking at. So putting this all together, I mean, it's clear that O-glycosylation is a key substrate recognition determinant in these O-glycopeptidase. And what's more to the point is that differential recognition of different kinds of glycans seem to be providing specificity to these enzymes. And of course, this makes sense because there's a variety of different core O-link glycans that these enzymes could potentially encounter. And these then again would all have their own sets of elaborations. And so we felt that what we were seeing was that variation in what we called those G subsites, where the glycans are being accommodated, were imparting the different specificities to these 3 different M60-like proteases. And so we could think of this on a larger scale now and extend this hypothesis to where we have this enormous family of peptidase M60 or M60-like sequences, and there's a great deal of sequence diversity. And possibly that the sequence diversity in this family reflects a variety of different active site architectures that mirror the multiplicity of possible O-glycan structures. And this is where we've been going recently. And we decided to tackle this by sampling a lot more of the sequence space. So we're now working on a large number of these enzymes. And what I want to do is focus in on a subset of them that all originate from the same organism. And this organism is Bacteroides cacti. So this is a gut microbe, and it's known for being an accomplished mucin degrader. It's armed with a wide variety of glycoside hydrolases, but these M60 peptidases are expanded in this particular microbe. So there's 16 different M60 peptidases found in the genome, and these are distributed between 13 different polysaccharide utilization loci. And we thought that we would select some of them. We couldn't do all of them. We'd select some of them and do this on the basis of maximizing sequence diversity. So we're kind of trying to kill 2 birds with 1 stone here. We're trying to study what the impact of the diversity in the sequence might be on substrate specificity, while at the end of the day, potentially getting more information about how this particular microbe is capable of degrading mucin in its normal environment. And So we chose to start this with bovine submaxillary mucin as our general substrate, and just briefly we chose this because there is a wide diversity of O-glycans on this particular type of mucin. Most of the core structures are represented, but it's not crazily glycosylated. There's no huge polylactosamine chains or anything like that, so it makes it easy to deal with. And we adapted a mucinase assay in my lab where we could quite quickly screen in a plate-based assay the activity of these enzymes. And so you don't really need to worry about how we name them, but the 12 are listed at the bottom here, and it's all relative to this no enzyme control. And we included plus and minus EDTA because EDTA should kill the activity. And so really, it's a bit counterintuitive, but what you're looking for is a reduced signal in the enzyme or the dark gray bars, and this will indicate likely activity. And so all of these, with the possible exception of BCM60B, showed clear activity on BSM. So this was great, good start. We could then start expanding this and using our defined approach to drill a little bit deeper into the possible glycan specificities of these enzymes. And this is where Warren's group, and particular Leaf, has done a lot of the synthesis for this. And so we did this screening by mass spectrometry on peptide sequences that are based on a MUC1 sequence. And so that's the MUC1 sequence right there. And it's important to note that there was no activity on the naked peptide, meaning in the absence of the glycosylation, there's no activity. And what you can see quite quickly is that these enzymes are not all created equal. About half of them showed some kind of activity, and the ones that showed activity didn't do the same thing. So for example, BCM60A is a specialist for this TN antigen, whereas BCM60G is a bit of a beast. It's active on everything that we tested. BCM60C seemed to be inhibited by the beta-1,3 linkage, but this is restored by the addition of the beta-1,6 linkage in the core 2 structure, suggesting something really interesting about what this particular enzyme might really like. And so overall, this continues to support this concept that these active site architectures are different, making them recognize different glycans. But there's a bit of a wrench in the works here, and that is, note some of them weren't active, okay? But they were active on BSM. And to be totally frank with you, initially I was a bit biased by our hypothesis that was, is it the glycan specificity here? Maybe our peptides didn't have the right glycans, and that's why they weren't active. And so this was actually kind of exciting at first, but some insight into this actually came with BCM60K, because we solved the structure of this relatively quickly. And it has an intact O-glycopeptidase catalytic site with a lot of the conserved residues that would recognize O-GalNAc. So check, it's definitely an O-glycopeptidase. And so we took the same approach as we did with ZmpB. So the bovine submaxillary mucin, a known substrate, was obliterated with IMPA, which is an enzyme that we call the beast. And then we soaked the crystal in that mixture of glycopeptides, assuming that this would pull out whatever it wanted. To our surprise, we picked out something that we were able to model, the T antigen, but most likely what is really here is a mixture of partially occupied TN and T antigen in there. And so we followed this up with a series of very specific experiments where we used glycoamino acids. And this is an example of just the T antigen, seronyl T antigen. So this protein definitely is active on BSM. It pulls out fragments from digested BSM. It binds glycosylated amino acids, but it's not active on the MUC1 peptide. And so we reason that this must be an issue with the amino acid sequence. And so this is, kind of a bit of a surprise to us. And so this is hot off the presses. Ben just did this yesterday. And what we did is we ordered a series of commercially available glycopeptides from Sussex Research. And we just assayed BCM60K on these and looked for activity by TLC. And what you can see here is this is the MUC1 peptide, but it's different than the one we used. It still has the same sequence, it's just longer, but there's no activity on that. So totally consistent with what we'd seen before for this particular enzyme. However, when we use the doubly glycosylated version, it's the same peptide, but the second threonine is glycosylated. We got activity. We got activity on the IgA hinge peptide, MUC5AC, etc. no activity on this one. So we were getting activity on glycopeptides. So, okay, well, that definitely seems to support the idea that amino acid sequence is important. What is difficult here is there's no real pattern that we can see, other than to say in what would be the P2 subsite, or sorry, the P2 residue is a glycine in both of these cases. Now, I totally admit that, you know, there might be a problem here that there's not much on the end here for an enzyme to grab onto potentially, but nevertheless, this is really the only shared feature that's obvious between the peptides that are active versus inactive with this particular enzyme. Overall, though, where we're headed with this then is this really complex set of substrate requirements, where we have these glycan binding sites which vary between enzymes, but now we have this idea where the peptide sequence does seem to be important, and you can imagine that this begins to make the order of complexity orders of magnitude worse when we're studying these. It's because now we've had to revise our hypothesis that both peptide sequence and glycan structure influence O-glycopeptidase specificity. And so these studies are ongoing. We're working on designing higher throughput approaches to assay the enzymes, which would then assist in being able to tease out these things. So if that wasn't complicated enough, though, I'll return to this idea here, where these are big proteins in general. And with respect to ZmpB, all I talked about was this catalytic module, but of course there's all this other stuff here. This is only about a third of the whole protein. IMPA, what I talked about, the enzyme is attributable to only this orange region. There's all this other stuff in this enzyme. And so, you know, what does this do? So with IMPA, we kind of got insight into this by accident. We made a catalytic mutant of IMPA, and we were soaking in glycopeptides to try and trap a Michaelis complex. We were really disappointed when we failed to ever get any electron density associated with the catalytic module. But imagine our surprise when we found clear electron density for the glycopeptide associated with this purple domain. And when we modeled it and looked carefully at this, what we saw was this proline that was sticking down into what I call this aromatic crucible. It's literally like a bowl made by 2 tryptophans and 2 tyrosines. And I've shown it in this angle here because this is a motif that's actually common to specific proline binding domains. But what do we also have here, really interestingly, is this glycosylated residue that precedes the proline that sits in the crucible. And this glycosylated residue with its sugar here, the sugar wraps around a tyrosine. And if we see it over here, it makes what's quite classic carbohydrate aromatic amino acid interactions. So this is providing evidence of the structural evidence of this really interesting binding motif. Overall, when we look at this and we look at the orientation of the peptide, this is completely consistent with a peptide being fed through the active site, which invokes this concept of multi-point attachment of this multimodular protein, where you have attachment here and here potentially to a substrate at the same time. Now, this concept becomes even more elaborate and more profound when we talk about ZmpB. We had structures of fragments of this overall enzyme for many, many years. And more recently, we revisited this in the lab. Ben Pluvinage made some more, let's say, refined constructs of large fragments, 2 large fragments, and miraculously managed to crystallize these things. And I say miraculously because multimodular proteins like this are just usually a nightmare for crystallographers. And crystallized them and got low resolution data sets. But armed with all this other high resolution structural information, it was very easy to put together an overall structure of this protein. And we could actually go and verify the structure using small angle X-ray scattering, which helped show that actually there's really not much flexibility in this protein. In other words, this model is actually a fairly reliable model of what this protein would look like in solution. And then this was supported by additional functional analyses of the carbohydrate binding modules, where 2 of the domains or modules were confirmed to have specific carbohydrate binding activity, with 2 more postulated to have carbohydrate binding activity. Now, this isn't the most useful view. We can turn this around a little bit, give it a surface, color the binding sites and the active site. And what's important here is that these all fall in a line. And for you that are watching this, these binding sites and the active site all fall on the plane of your screen. So they're all in a nice line. And the reason that this is relevant is somewhat revealed by some recent low-resolution cryo-EM work on mucin domains. And so that's shown here. So this is a cryo-EM reconstruction of mucin domains from Uc5b. And what was observed is a highly repeating pattern of these domains that are roughly 50 nanometers in diameter, but also separated by 50 nanometers. So they're on 50 nanometer centers. And so these blobs were postulated to be the highly glycosylated regions, with the intervening regions slightly less glycosylated. What's important here is that this protein, all of the CBMs are on 50 nanometer centers, 100 nanometers between these, which really invokes this idea that this protein is designed to, or not designed, but it has evolved to sit down on these regularly repeated domains of a mucin. This is on a fraction of that 50, which would place the catalytic activity in between these domains. So it's a really tantalizing picture about how this really large, architecturally complex protein is customized to degrade mucin. So by way of summary, where we're at here now is we have this really diverse family of proteins that all show some, or at least so far, all show some specificity for glycans in their substrates. But also now we're seeing some potential for amino acid sequence specificity as well. And so given the diversity of it, this really invokes this curious idea of restriction enzymes for glycoproteins, possibly. So that's something that we would investigate. And of course, these enzymes are now finding quite regular use as tools in glycobiology, where people are using them as mass spec tools to study glycoproteins. Now, of course, we have this additional layer of complexity where the ancillary modules that are present, really common actually in these M60-like enzymes, provide this additional layer of being able to target a protein to its glycosylated substrate. And so with that, I'd like to thank all the players in this. Particular shout outs to Ben and Ashley Elite and Liz, who more recently Ben and Ashley have been driving this, and historically Elite and Liz. And then of course, Warren's lab, they've all been very dedicated in generating these glycopeptides, which have really been important in vaulting this research forward. And then of course for funding, now the interesting thing about this, and the reason that this research has been going on and off, is that this project has never been formally funded. It's always been sort of offshoots of various different projects that we've had going, which we've managed to keep this ticking over. But still, I'll thank CIHR and GlycoNet for that funding. And with that, I will entertain questions.